Next, let's talk about titration of a polyprotic acid. Polyprotic acids have more than 1 Ka value, so that means your curve is actually going to have an equivalence point for the removal of each proton. So here, if you have sulfurous acids, this is a weak acid, it has 2 protons. So the first thing it's going to do is it's going to lose its first proton, and there's its Ka, so here, and then its second equivalence point, you're going to take this and it comes down, and then you're going to take the proton off of it, and you're going to have your second equivalence point. Now, if you were to run like just a titration calculation, you would calculate that it takes the same amount of base to remove each proton, but the pH difference is going to change a lot. So you're still going to have your, your little buffer region for each one of them, your flat portion. And this is something that we'll actually do in the lab and try to get it constructed. But you can run calculations there. So your first equivalence is going to be half of your total volume of base, and your second equivalence is going to be the other half. So that's what I was demonstrating on the graph. So let's say it in words. So if it takes 25 milliliters to get to your first equivalence point, then it's also going to take 25 milliliters to reach your second, and we get that according to stoichiometry.